Bismillahirrahmanirrahim. Ji, out of unit number 3, which is biological molecules, now the 3.4 portion is to discuss carbohydrates in details. So let's start with the proceedings. Carbohydrates contain only the element carbon, hydrogen, and oxygen. Rather the first document about the biological molecules, we have already discussed that the definition of biochemical definition of carbohydrates is that these are the compounds having polyhydroxy, aldehyde, or ketones, are the compounds having larger molecules, but on hydrolysis, they may give rise to those chemicals having polyhydroxy, aldehyde, and ketones. So that was the definition. Now we are supposed to discuss carbohydrates in much more detail. So carbohydrates contain these only three elements: carbon, hydrogen, and oxygen, and they can be grouped into monomers, dimers, and polymers. Sometime we may use another category, which is oligosaccharides and having more than 2 and less than 10 or about 10 monomers. So this is the division of carbohydrates. Simply, they may be named as sugars because they are sweet in taste. So out of these, monosaccharides and disaccharides are sugars, but the polysaccharides, they are not sweet in taste, so they are not named as sugars, simple sugars. So monosaccharides mean one unit, monomers. For example, glucose, fructose, ribose, and we may categorize glyceraldehyde, dihydroxyacetone if we want, but they are not precisely monosaccharides. So disaccharides are having two units, dimers. So the well-known examples are sucrose, maltose, lactose, and then the polysaccharides, which are having more than what we say 3 units of monosaccharides. So they are polysaccharides. But generally, the compounds having more than 10 and so on monosaccharides, they are named as starch, cellulose, glycogen. So they are polysaccharides. So what are monosaccharides? So they all have the formula CH2O whole N, mean that maybe the repetition of these units. So where N can be 3 to 7. So they are monosaccharides. The most common and important monosaccharide is glucose. We know that this is a 6-carbon or hexose sugar, and its formula is C6H12O6. So its structure is quite like this. So the dark lines show that these molecules, they are facing us, and the rest of the molecules may be it may be hanging like that. And the darker area, if we see this darker area is facing our side. So if we omit the carbon numbers, so the shape may be more simpler. So glucose forms a 6-sided ring, but its structure in water is like a chair-like structure. So it is beyond the scope of this syllabus. So no need to discuss this thing. The 6 carbon atoms are numbered as shown: number 1, 2, 3, 4, 5, 6. 6 is out of the ring, as we can see from this side. So this one is 1, 2, 3, 4, 5, and 6th carbon of the glucose molecule. So there are some isomers of it. For example, fructose, galactose. So fructose is again 6 carbon, but it is a ketone. So if we have the ring-like structure, so it is a 5-ring structure. So common 5-carbon sugars in which carbon number is 5. So they are ribose and deoxyribose, which are present in DNA and RNA respectively. So they are 5 carbon. So disaccharides mean that the compounds which are made up of are having two monosaccharides which are joined together by glycosidic bond. The bond between two monosaccharides to form a disaccharide is glycosidic bond. So, and water will be formed in this, and this reaction is condensation reaction. So, what is like that? This is one glucose molecule, and if we add another glucose molecule, so from carbon number 1 of first glucose and from carbon number 4 from of the second glucose molecule, there will be formation of water, and water will get released. So we will get this glycosidic bond in this case. So this is a disaccharides. So this shows that two glucose molecules join together to form disaccharide, which is a maltose in this case. So because this bond is between carbon number 1 of one molecule of glucose and carbon number 4 of other molecules, so this is alpha 1,4. Why it is alpha? Because in this case, the position of OH group is lower. So that's why it is alpha. If it is above the carbon atom, then it is a beta position. So this kind of reaction in which water is gets released from the reaction, after this reaction is condensation reaction. So reverse of this is hydrolysis, because water is getting added in it and releasing the two monosaccharides. So in general, polymerization reactions are condensation and breakdown reactions are hydrolysis. So there are three main disaccharides, maltose, which is given in above example, in which glucose 1,4 linkage is there between two glucose molecule, glucose 1 and glucose second is sharing carbon number 4. So this compound is being released by the amylase enzyme due to the digestion of starch. We all know that brewing beer starts with malt. We get this malt from the barley, the germination of barley seeds, so which is maltose solution made from the germinating barleys. So maltose is the structure shown above. So sucrose, cane sugar, or table sugar. So in which the bonding is between two different monosaccharide. One is glucose, another is fructose. So bonding between glucose carbon number 1 and fructose carbon number 2. So it is common in plants because it is less reactive than the glucose, and it is a main transport sugar released after the formation of after photosynthetic reaction, and will be transported through xylem to the storage organs or organ where it is being used. So it is the common table sugar. So we put in our tea. So when we say sugar, it means sucrose. When we name it as table sugar, it means sucrose. So lactose is milk sugar. It is generally naturally present in glucose, in which lactose, one monomer is lactose and other is glucose. So lactose is sharing its carbon number 1 and glucose sharing carbon number 4. So it is found only in mammalian milk. So main source of energy for infants, infant mammals. So then comes the polysaccharides. So polysaccharides are long chain of repeated monosaccharides. Once again, bond is glycosidic bond. So in our syllabus, there are three main starches, polysaccharides are there. Number one is starch. It is a plant storage polysaccharide. It is insoluble in water and not sweet in taste and forming granules inside the plants. What we say, the plastids. So it is not causing any disturbance in water potential because it is not soluble in water. So as we are supposed to, and we should mention it, that starch is not a single compound. It is having two units, amylose and amylopectin. Amylopectin is simple poly 1,4 glucose, it is straight chain. So chain is slight, can be feedback around, rollback. So amylopectin is 1,4-glucose, but out of total units, glucose units, so about 4% they are having branches, 1 to 6 branches. So here you can see that the branches, this was straight chain, but from this carbon, this glucose onward, there is 1,6 linkage, and once again remaining linkage is 1,4. So wherever we are having certain sort of link, the side branches, so they are having 1,6 linkage. So it is a quite complicated and complex than the amylose. So amylopectin is the part of the starch. So both are contributing towards the formation of starch. So then comes the glycogen. Glycogen is similar in structure like the amylopectin. It is poly 1,4-glucose, but the branches is more in frequency, 9% of total, what we say, polysaccharide glycogen. There are certain branches, 1,6, 1,6, 1,6. So it is a sort of a complex molecule as compared to simple starch. And it is the polysaccharide which is being stored in our liver and then muscles. So can be used after its requirement when glucose is depleted, and then glycogen will be converted into once again glucose to form and to maintain the glucose level in our body. So cellulose is only found in plants. It is poly 1,4-glucose, but there is one difference, that the bonding is different. In this case, the starch and glycogen contain alpha glucose, in which hydroxyl group on carbon number 1 stick down, as I have already mentioned, from the ring. But in this case, cellulose contain one beta glucose, and its carbon number 1 is sticks up. It is up in position. So, for example, in this case, if we look for this combination, starch is having alpha 1,4 glycosidic bond between two glucose molecules, but in this case, there is beta bond between 1,4 glycosidic. It's a 1,4 glycosidic bond, but this bond is beta because this is between OH groups. So they are above. So at least one is having this one, and the other is sharing like that. So glycosidic bond in cellulose is different. So this molecule is having many numerous glucose units in it, and they are forming microfibrils, and microfibrils, they combine together to form cell wall in this case. So in between different chains, there may be hydrogen bonds due to the difference of electronegativity between oxygen and hydrogen. So beta glycosidic bond cannot be broken down by amylase, alpha amylase, which is present in our body, but it can be digested by bacteria and certain other microorganisms, and they are present in herbivorous animals like cows, rather cattle animals. So they are using and digesting this cellulose due to bacteria, symbiotic bacteria, or and they are very what we say useful for their food nutrition. So as humans, they are not able to digest cellulose, so it is referred to as fiber because it will providing some other benefit to us. So it is roughage or fiber, and causing some smoothness in our smoothness of peristalsis movement in our alimentary canal. There are some other polysaccharides like chitin, pectin, agar, murin, lignin. So chitin polyglucosamine having one extra, what we say, this amine group along with the glucose, along with the monosaccharides. Pectin, it is composed of polygalactose uronate compound found in plant cell walls. Agar polysaccharide, polygalactose sulfate found in algae. and it is used in what we say in our culture as a stabilizing agent, solidifying agent in our experiments, and our different puddings and other uses are there. So murin is a sort of sugar peptide polymer found in bacterial cell wall, lignin, a complex polymer found in walls of xylem cells, and in main, which is a main component of wood. So that is about different units of carbohydrates, and we are supposed to distinguish them because we may get the MCQs in our exam about these things. So the syllabus is this. So this is all from my side for this lesson. Thank you very much. Allah Hafiz.